Good afternoon and welcome to the first of 3 virtual lunch speaker sessions in 2020, hosted by the Northern California Real Estate and Construction Council and made possible thanks to sponsors Langen, Perkins & Will, TMG Partners, and the San Francisco Business Times. I'm Chris Robbins, and I'm proud to represent the Northern California Real Estate and Construction Council as board president. This session will be recorded. I'd like to welcome many of our hardworking volunteer groups, the Northern California, Los Angeles, and Chicago Real Estate and Construction Councils. These councils are comprised of developers, brokers, contractors, building owners, engineers, architects, and consultants who have a single philanthropic goal: supporting the life-saving, treatment, and compassionate care of City of Hope. To date, these 3 real estate councils have raised over $55 million. I'm also honored to have many other volunteer groups joining us today. Thanks to all of you, our volunteer event fundraising tops $40 million annually in support of City of Hope's work. While we usually gather in person at our annual events, such as our Spirit of Life Award Dinner and Bike to Hope events, we are grateful to at least come together with you virtually today. And honored to provide you with unique access and an in-depth view of the incredible science City of Hope works on day in and day out in their fight against cancer, diabetes, and other life-threatening diseases. For those of you new to City of Hope, I'd like to share a few facts. City of Hope was founded in 1913 in Duarte, a city located in Los Angeles County, originally to treat patients with tuberculosis. Over the years, the 10-acre campus has expanded to over 110 acres, 30 additional outpatient centers in Southern California, and collaborations with hundreds of the top cancer centers globally. For over a decade, City of Hope has consistently been deemed one of the nation's elite cancer hospitals. They conduct hundreds of clinical trials, with their research resulting in 4 of the leading cancer drugs that are being used all over the world today. And now I'm proud to introduce today's guest speaker, Dr. Yuman Fong, a truly remarkable man I've had the privilege of getting to know over the past 6 years. A pioneer in both the operating room and in the laboratory, Dr. Fong, the San Giacomo Family Chair in Surgical Oncology, in addition to the chair of City of Hope's Department of Surgery, is an internationally recognized expert in cancer of the liver, pancreas, gallbladder, and bile ducts, and leads the research effort to employ genetically modified viruses to destroy cancer cells. Dr. Fong joined City of Hope in 2014 after more than 2 decades at the renowned Memorial Sloan Kettering Cancer Center in New York City. He is both author and innovator and has developed many new surgical techniques and instruments and written and edited hundreds of scholarly articles as well as nearly a dozen textbooks. In normal times, as Dr. Fong's extended family lives in San Francisco and New York, when he is not visiting family, operating on patients, or conducting life-saving research, he and his wife Nicole can be found ballroom dancing, traveling to NASA rocket launches, flying up for Warriors games, and enjoying concerts any night of the week, like seeing Blondie on a Sunday night before flying to San Francisco for our NorCal real estate industry's honorary breakfast last year. Welcome, Dr. Fong.
Thank you very much, Chris, for that kind introduction. So today I'm going to talk a little bit about cancer care in COVID-19 times. And the reason I'm going to incorporate COVID-19 into this is because it's hard to get away from it, because it's all around us. It actually has influenced all aspects of our lives. And now we need to go and decide how we're going to go and lead our lives within these times, as well as try to figure out how are we going to go and take care of patients during these times, okay? But just a added introduction to the City of Hope. This is a picture of the campus. We're a suburb in Los Angeles, okay? People always ask me, where's Duarte? Well, we're just a suburb on the edge of Los Angeles, and therefore, again, I invite all of you to come and visit us when the COVID-19 has passed and it's safe to travel. And we are 130 acres on the outskirts of Los Angeles, and we've been delivering amazing cancer care for a long time. But let me tell you some of the other things that we have done. This has been a cancer center that's been in existence for about 100 years. And when tuberculosis, which was our first mission, I fell by the wayside because of antibiotics, we actually went ahead and changed and pivoted and started studying diabetes. And in 1978, this was the place where artificial insulin was invented. And because of that, really changed how patients with diabetes actually took care of themselves. Then in 1983, we went ahead and invented humanized antibodies. Those are antibodies that have become many, many medicines for cancer patients and for various types of patients, and really change how modern cancer care actually happens. Avastin, Herceptin, many of the other life-saving medicines actually are able to be delivered because humanized antibodies were enabled. It was one of the first places to do bone marrow transplant, and therefore there is now over 12,000 patients treated with bone marrow transplant. And in America, this is the safest place to have a bone marrow transplant. But because of the history of innovation, it was also one of the very first places that did robotic surgery, and that's where I intersect with it. And that's because I do robotic surgery. We've done over 13,000 cases here on campus, and we are one of the leading centers in robotic surgery. And as Chris has said, we are now the highest ranking cancer center by the U.S. News and World Report, and we are #1 in the West. Just to start here, and that's because all our lives are really been influenced by COVID-19 in the last months. This is what it looks like. This is the virus, and the reason it's called coronavirus is because all these spikes. Okay, and that's what gives it its corona appearance, and that's why it's been named that. But in reality, what it is an RNA virus. It's a big virus, and it has 30 kilobase pairs of genetic material, which makes it a big virus. And just for all of you that help with the homework of your children, or plan on going on Jeopardy, I just put down the genus, family, order, and realm of this virus in case you need it. But basically, this virus can infect many things in the body, but mainly infects epithelium of the airway tract. Okay, so from the top of the nose all the way down into the bottom of your lungs. Okay, and that's where it really likes to live. We're going to talk a little bit about viruses later for cancer therapy. And it's very clear that viruses only like living in certain things. And COVID-19 virus likes to live mainly in the lining of the lungs and airway. And that's why we test for it by swabbing the back of the nose. And that's why we look for it and look for damage from it within the lungs. But it's not just the virus infecting the lungs and making many more viruses infect neighboring cells that make it dangerous. This particular virus also triggers all of the body's immune cells, whether they are neutrophils, monocytes, T lymphocytes, or macrophages. These are immune cells that normally protect us, but when they react to this virus, they produce many, many cytokines. These are hormones that actually are immune hormones that try to go fight the virus. But when the fight is too vigorous, it creates terrible damage within the lungs and within other organs of the body. And part of the reason it's so dangerous is that in certain individuals, this immune reaction actually turns out to kill the patients. I want to show this very sobering slide, okay? March, this is what was happening in the world, okay? Here's United States. This is in the green. It was very much under control in the beginning of March, okay? The number of cases that were being discovered compared to other places on Earth, including Italy and South Korea and Iran. We thought we were in great shape, and we were. And then now, okay, and here's in the United States, okay? We are #1 in the world in terms of number of cases now, over 3 million cases, over 3 million cases. And it was less than 3,000 cases back in March, okay? So the climb here has been really steep, and we've got to do something to control it. Otherwise, our entire population is going to be of worry. Okay, we have actually passed everyone else in terms of total number of cases, And there are very simple things we could do to protect ourselves. And so the public health response, and this is what everybody's been trying to do. First of all, do the lockdown, okay, so that we can actually make sure that the cases of infected patients remain only within the infected and don't bring new cases. Wear personnel protection equipment. That could be mask and eye protection. I'm going to talk a little bit about that. Testing. So knowing who's infected is important because it turns out, depending on the age group, a big percentage of patients don't show symptoms. So if you are between 20 and 30, even if you caught the virus and now you're making more virus and you can actually spread it to other people, 99 times out of 100, you may be feeling just fine, okay? Whereas if you are over 80, the chance that you are going to be sick from it is very high, and the mortality rate approaches about 30%. And therefore, how do we go detect for the virus and protect patients against those individuals that are infected, can spread the virus but don't feel sick, okay? And then later, how do we go test for antibodies so that we know who's protected? And we're going to talk a little bit about that. And then once we actually have it under control, that we know who has it, we are protecting those patients that don't, people that don't have it and are particularly vulnerable to disease from it. How do we emerge back to normal life, okay? And we were all hoping that by this point in the year, we would be going back to here. But unfortunately, for various reasons, we are still stuck here, okay? And the cases are getting worse. So people always ask, you know, should I wear a mask, okay? And there is no doubt that a mask is helpful, okay? And so let me explain why, okay? When you think about the virus really exists in two forms. It could be the virus by itself, in which case it's a very small particle. It's about 1 micron in size, teeny, teeny, tiny, okay? And when it's in this form, it could be aerosolized, meaning it could actually go into the air. If somebody sneezes and they actually have the virus in the back of their nose, it's going to get into the air in these circles here, and it could actually travel a long, long ways, and it lingers in the air in whatever room it was, okay? So the person could actually leave the room, and this form of the contagion, the virus by itself, can still be up in the air. So time in the air, I'm sorry, this is the reverse. This would be minutes, okay? Alive on surfaces, hours. Alive on animals, it's days. But again, when it's in aerosolized small form, it's actually up in the air for many, many, many minutes, okay? And it could travel a long ways. The other form of the virus is really when it's actually combined with fluid in our airway. So let's say somebody sneezes and the mucus in their nose actually is caught up in the virus. Suddenly, it's really a clump of mucus with many virus on it, but it gets very big. It gets to be about 30 microns in size, and those particles are called droplets, and they're much bigger than the virus itself, and those fall down usually within 2.5 meters. That's where the 6 feet comes from, okay? That's because if somebody covers their mouth when they're sneezing, such as with a mask or with a handkerchief or with something, and they do a big sneeze, usually droplets come out, and then within 6 feet, it drops to the ground. And as long as you're not touching the ground and touching your face, the area becomes safe pretty quickly. Whereas if it's aerosolized, without a mask, without a handkerchief, without something that slows down the virus and allows the small form of the virus in the air, it travels a long, long ways, okay? And that's why having folks without masks who are with active infection makes a whole room at risk. And whereas as soon as people put on their mask, suddenly everybody is much more protected. So wearing the mask is really protecting our neighbors, okay? And protecting our friends, protecting our family. But protective equipment is not just a mask, okay? And that could be a regular mask, surgical mask, 3 layers. The reason we put 3 layers in it is because, again, the virus is 1 micron in size, and the pores in most thick, dense cloth. So if you take a thick, dense cotton, the pores are still about 3 microns in size, okay? And therefore, again, if you just have 1 layer, it has a chance of coming through. If you have 3 intersecting layers, then suddenly the chance that aerosolized virus in a small form gets into the air is very low. N95s are even tighter, and N98s, and here's a P100. But it's also protecting your eyes, okay? Because if it's really an aerosolized form in the air, or if it's droplets and you're standing nearby, it can get on the mucous membranes of the eyes, and that's why people are wearing either goggles, face shields, but now there are many companies making very, very attractive goggles that can protect you, and most of those are motorcycle accessory companies, where most motorcyclists have been wearing goggles for a long time, and a lot of those are applicable to protect you in your travels. If you get back to traveling, it would be a good idea to look at some of those companies, and if you need recommendations, please email me, okay? But wearing gowns, wearing this, which is a PAPR. This is a complete protection where the air is actually pulled in through a HEPA filter, so it filters out the virus, and only fresh air gets into the tent, so that this is what people wear when they're actually actively taking care of patients who they know have COVID. And but again, these are in enormously short supply, and that's been one of the problems. And here are the gloves that go with it, okay? So at the City of Hope, in my department, very early on, when there was huge controversy about whether people should wear masks at the workplace, we wrote a very important paper that for wearing masks for surgeons, where we actually broke this down, and it's been one of the most downloaded articles on the internet in terms of looking at wearing masks in this crisis. And so again, protecting each other is really important. How about treatments, okay? There have been a lot of things touted for treatment of COVID-19, and what we do know is that remdesivir seems to work. This was a drug that was actually originally designed for Ebola. It didn't work so well for Ebola, but some of the enzymes in the virus are the same, so it was tested in COVID-19, and it actually reduces mortality of patients that are on ventilators. So now it has received rapid approval by the FDA for use, and this is actually the most important drug when somebody's actually on a ventilator and very, very sick from COVID. And anti-inflammatory drugs seem to help, so steroids, dexamethasone is given now routinely for patients who are very sick. But there are also a class of hormones that come from our immune cells that are responsible for hurting us by having this overly vigorous immune response, and that's why having agents that block it, so interleukin-1, interleukin-6 are some of these hormones, and drugs that block interleukin-1 and interleukin-6 are now being tested and being used in COVID-19. But I'm sure you guys have heard of the fact that some patients recovering from COVID-19 are known to have good antibodies in their blood, and therefore, if a patient is really getting sick from COVID-19, finding a patient who's recovered of the same blood type and transfusing just the plasma from one patient to the other actually can help, okay? And then now there are enormous networks set up all through the United States to go match patients with people with their same blood type who've recovered from COVID-19 to get convalescent plasma for transfusion. And at the Mayo Clinic, there's a great big network that's set up. But at the City of Hope, we just got $1 million grant from CIRM, which is the California Stem Cell Initiative. And we are actually have set up a whole network in California to go match patients with convalescent plasma to make sure that everybody gets the best chance of survival. But there are other treatments available, and they include anticoagulants and antibiotics to support people through from the, because if somebody who has COVID-19 also has blood clot problems, and many of the small blood vessels in vital organs get clotted off, and by just using anticoagulants, we can actually prevent that and preserve organ function. And then many organs actually get bacterial infection on top of the viral infection, and giving antibiotics in those situations seems to help. The area that people have talked about the most, though, is really vaccines. And more than 150 companies and academic groups are now trying to produce a vaccine, okay? We actually have 2 separate groups at City of Hope working on it. And but for the vaccine to work, these things have to happen. The first question we have to ask, will it produce immunologic reaction? So that means everybody talks about the antibodies. Again, when somebody has an immune reaction to something that is foreign to them, antibodies are formed. Those are through something in an immune cell in the body called the B cells. But it's also that there's a kind of cell in the body called T cells, T lymphocytes, that actually attack things that are foreign to us. So it would attack bacteria, viruses, or even cancer. And again, making sure that the T cells recognize COVID-19 is going to be a very important thing ongoing. And so the good news is that a number of companies now have proven that they have designed vaccines that could produce an immunologic reaction both in animals, and usually they're in primate kind of testing, and some have proven that they've done this in man, okay? And so the publication earlier this week on Monday in the New England Journal from the company Moderna certainly shows data that they have a vaccine that could produce an immunologic reaction. But will the antibodies and the T cell memory last? Okay, that's going to be one of the things that's going to be crucial in this. And there's now growing evidence that in many individuals, being vaccinated or exposed to COVID-19 does not produce long-term immunity. So I'm going to come to that. Will antibodies and T cells protect against the infection? So just because you have antibodies that recognize the virus and T cells doesn't mean that you will be protected. So when people get cold sores, okay, cold sores come from a virus called herpes simplex. 90% of Americans actually have antibodies, but many people still get cold sores over and over again, and it's because, again, those antibodies didn't protect completely against it. And if we had antibodies and T cells that protect against infection, can we make enough doses of a vaccine for human use? That's because, again, in America, we will need to make 250 million doses plus. For the world, we will need to make 7 billion doses of vaccine. That's a huge undertaking that currently there is no infrastructure in the world that currently has done that ever for a single medicine, okay? And so will we be able to do this for it? So again, many challenges coming up, some steps forward that are very encouraging. So I'm hoping we're going to get to a place where we're going to be able to have a vaccine for this. But the sobering thought that I want to leave you with is that we've been trying to make an HIV vaccine for 30 some odd years, okay? And we do not have one. What evidence do we have that maybe somebody immune is not going to keep the immunity? So in a study in Nature Medicine from June, this group from England, they looked at patients who had been infected by COVID-19 and looked 8 weeks later to see how many of them had become seronegative, meaning the antibody disappeared, okay? And that was 40%. So it makes us very worried, okay? That the antibodies that is produced by 8 weeks might be gone in a significant number of individuals. Sal looked at this for patients that had COVID-19 who had potent antibodies before, and only 17% of those maintained it after 2 months, okay? And thirdly, this is a very sobering study that just came out in June. They looked at 3,832 healthcare workers in Wuhan and measured their antibody levels, and only 4% had antibodies. This was the healthcare workers that in the first wave of infection, when it overtook Wuhan, took care of patients, we would have expected that the majority of these folks were exposed, and that if the antibodies were long-lasting, that we would see it in most of these individuals, and only 4% had it. Now, doesn't mean we shouldn't go ahead with these vaccine strategies, but I think we're going to need more data before we think that's going to be completely protective. And so how do we go and go forward? Big questions coming up, okay? What did we do at the City of Hope? At the City of Hope, March 15th, when it was very clear that COVID was coming to America and coming in a big way, we shut down the campus and canceled all elective surgeries, okay? And that's because the goals were to provide world-class cancer care while protecting all of our patients and staff from COVID-19. Because we only do cancer, all of our patients are vulnerable, okay? They are all at high risk from dying from a COVID infection. So March through April, we ramped up testing, okay? And by early April, we were testing every patient coming for surgery. And by April 21st, we were testing every patient coming into the hospital. Since then, we've done over 6,000 tests and 44 outpatients have tested positive. We've prevented those folks both from getting surgery or coming into the hospital if they're asymptomatic. And then I'm going to explain why it was important to find these people and not do surgery on them if they needed surgery immediately. Because just waiting 14 days, all of the patients recovered from this and could come to safe surgery. I'm going to come back to that. All the cancer patients that we were treating that developed infection in the community, we actually admitted them to a different building at the City of Hope, and therefore at our main campus hospital, we still have not had a single case of COVID-19 in the main hospital. And what that has allowed us to do is ramp back up on mission, and that mission is cancer care, and that's because as soon as it spreads into the main hospital, very, very difficult to go take care of very vulnerable patients with cancer, okay? Now, as the chief of surgery, one of the first questions I asked was, is it more dangerous to operate on somebody who had COVID-19, okay, active COVID-19? So this was a very important paper for us, and it is a paper came in the Lancet from Wuhan very early on. 34 patients undergoing elective minor, some of these were minor surgeries, and 44% of them had to be admitted to the ICU, and 1 out of 5 of these patients died. Okay, very clear. Since then, there's been a follow-up study from the UK that looked at the world experience of COVID positive patients undergoing surgery, and they looked at the multiple centers and pulmonary complication, meaning lung problems, 51%, and when they looked at 30-day mortality, 24%. Okay, some of these were minor surgeries. So we know that if somebody has the virus active, if we take them to surgery, give them anesthesia, do mechanical ventilation, we're probably hurting them. And so what we've been doing is that as soon as we test somebody positive, they are feeling well, we send them home to quarantine or wherever to quarantine for 14 days. We retest them, and everybody's gone negative at 14 days, and then we operate on them, and it's been very, very safe. And so that's why testing is important, okay? But cancer screening also becomes an issue. Taking care of cancer patients is not just what we do in terms of chemotherapy and surgery. It's finding the cancers and finding them as early as possible. I'm asked all the time, when are we going to have the cure for cancer? One of my answers is that we already do for a segment of the population. That's because most early stage cancers are cured. Okay, if somebody has stage 1 breast cancer, likelihood we cure them is above 97%. Okay, the trick is just finding cancers at early stages. Okay, so we can screen for cancer and find them early. We are curing a lot of people. And COVID-19 has slowed down screening everywhere. In the UK, it's estimated there are 2.1 million people waiting for routine screening. In the US, there was an 86 to 94% drop in mammography in the last 3 months, okay? And this editorial just came out in Science, and the thought was that for every 6 months delay in diagnosis, there will be a 1% increase in cancer mortality. So again, getting back to cancer screening, really important. Getting back to good cancer treatment, also important. I would urge you and your family and your friends, if you're due for a cancer screen, go find a safe facility, continue your health, because cancer still remains a high killer of Americans. So we've been returning to the mission of care, and the mission of care for cancer care really is great clinical care, innovative research to find effective new therapies. It's also rapidly moving laboratory findings through well-designed clinical trials of human therapy. So great standard care, innovations to find new medicines, and then moving those medicines to standard of care as quickly as possible. So I'm a kind of simple surgeon. I go to work every day saying to myself, going to do more cures, less invasive, okay? Because the other part of it is that how small an incision can we use? How little side effect can we have? Trying to make sure that we are less invasive as we get better is also really, really important, okay? So what I do in daily life and in the clinics is really do liver and pancreatic surgery. That's all I've been doing for the last 35 years. And the most common disease I treat is liver metastases from colon cancer. So colon cancer goes to the liver very often, okay? And that's because the veins draining blood from the intestines really is through a vein called the portal vein. And this vein brings all of the food that's been absorbed up to the liver for processing, because the main job of the liver is actually to process food. That's why cancers of the intestines, if the tumor cells get into the blood first place, it goes to the liver. And so half of the people who have colon cancer will eventually develop liver metastasis. Survival for untreated disease was 5 to 10 months, and 5-year survival, almost unheard of in somebody not treated, okay? So when I graduated from medical school in 1984, almost no one lived, because many of the patients we didn't even treat, thinking cancer cells in the blood and gone to the liver must be everywhere. It turns out the liver is a really good filter, so many times the cancer doesn't go anywhere else except the liver. Also turns out the liver is really good at killing cancer, so millions of tumor cells has to get to the liver for a few spots to actually form. And we started noticing this in the 1980s, and we started removing this stuff from the liver. And what we have discovered in the last 40 years, which is kind of cool, is that even though this is stage 4 disease, surgery can be curative in 40% of the people. We're now curing over 40% of the people, meaning we allow them to die of old age or some other disease, okay? And that is quite remarkable in a 30, 40-year period. In fact, now we're doing it in a very minimally invasive fashion. So let me show you a patient that came to see me not long ago. This was a physician from a different country who had a sigmoid cancer discovered. This is in the colon on a routine colonoscopy. And then they scanned him and found in his liver that there was one spot of metastasis, okay? So he called me and wanted to come to America for the treatment. That's because given cases like this, what I'm now doing is really robotic surgery. I actually reach in with the robot, and this is the robot standing on the side of the patient. I aim the robot one way and take out the liver tumor. Then turn the robot and aim it the other way, and I take out the colon cancer. This is what the robot looks like. This is what a typical patient looks like after surgery with tiny little incisions. And so he came. We were able to go do that for him. He was out of the hospital in about 3 days and back to his country within about 10 days, okay? And so these combined resections, it's the fact that we now can treat stage 4 cancer and provide potential cure. This is all huge steps in treating colon cancer, okay? And so the 170,000 people in America that go get colon cancer, half of them will have metastases in the liver. Now we have great treatments for them. So having pushed this field quite a bit, okay, and helped a lot of this move forward, what we have discovered is that we can actually now get people home very quickly. So again, the number of patients of mine, like this is the last 100 that I did, and most of the patients are home in day 1, 2, and 3. So some of these patients are actually, this is outpatient surgery now for some of these patients where they don't even come and stay in the hospital. And their functional recovery is quite good. We are now tracking all of these individuals. That's because if we send patients after liver surgery back home within 1 or 2 days, we actually need to be sure they are doing okay. So we now put this on them. This is a VivoFit. It's not a Fitbit. VivoFit comes from the Garmin company, and I get nothing from the Garmin company. And but this I use because it is a true healthcare device. It has where Apple Watches, and if you forget to charge more than 24 hours, it's dead. That's why an Apple Watch is highly unlikely to be a true health device, whereas health devices, sick patients can keep them on all day long. This is also waterproof, so they could get in the shower. So now I track what patients do at home, because we have to be sure people are recovering. And so now this is a typical recovery from a liver surgery, and they're walking 6,000, 7,000 steps before surgery. They are in the hospital for a day or two. They get back to normal into 6,000 steps very quickly. Within a week, they're back to normal in terms of function. And so again, what other trackers we move forward? And so Virginia's son, who is responsible for the quality of life and home monitoring program for us, we've been rolling out different methods of tracking people at home, and we are in the process of designing next generation devices to go help really to make sure patients at home are doing well. These are particularly important in the COVID times where people are afraid to come to the hospital or can't come to the hospital, and now telemedicine in terms of device-driven telemedicine really is the current really important area of study, and hopefully will be our near future state in taking care of these patients. But for liver resections, I went and looked in California state data recently. Okay, we looked between the year 2000 and 2011. The peach here represents how many cases of colon cancer. Liver metastasis is shown in green. The purple is how many people actually got liver resection. Okay, so the sad part is that half the people got liver metastases, but less than 10% got a liver resection. Okay, we estimated it should have been closer to 40% of these patients. We think that right now there's still a knowledge gap in the public, knowledge gap in our caregivers, and that it is possible to cure patients of metastatic disease in the liver, and we're trying to get the word out. So you'll see a huge number of papers coming out very shortly on why this is a big detriment for our patients, and we're going to try to engage with primary care physicians, oncologists, and with our state legislature to try to provide more individuals with a possibility of longer survival and potential cure. So moving on further, okay, my laboratory work is on designing viruses, okay? So let me tell you what this is, okay? On the left-hand side, this is a plate of a kind of cancer called mesothelioma. This comes from the lining of the lungs, and usually it's from cigarette smoking or asbestos. And I've thrown onto this plate one virus for every 100 tumor cells that's there. The virus is labeled with fluorescent green, so when the cell is infected, it turns green. And that's why over time, this is 13 hours, you'll see more and more of the cells being infected and turning green, and soon you're going to see it burst. And then within about 24 hours, all of these cells in this very, very resistant cancer, everybody who gets mesothelioma dies of it, okay? And it is a terrible, terrible disease for which we have very little treatment. And if you pick the right virus, you can infect them all and kill the cells. This particular virus is currently in human clinical trials in America for mesothelioma, and we're very excited about it. So let me introduce this field that my laboratory has been working on for 25 years, okay? So not all viruses are bad, okay? Viruses just like living in certain cells, and if it happens to be a human cell essential for life, it takes life. What we've been trying to do is to find viruses that don't like living in normal human cells, but like living in cancers, so that we can actually let the virus go in and infect cancers and kill them. So the whole field is called oncolytic viral therapy. And the whole idea of the field is that we want to engineer viruses that infect and kill only cancer, carry additional payloads to kill cancer, and those payloads could be checkpoint inhibitors, those are immunotherapies, anti-angiogenics that stop blood vessels from growing to feed the cancer. And the way that these cancer viruses work, it both directly kills the cell, it also activates the immune system to recognize the cancer as foreign, so that the immune system could kill more of it, okay? And this is not science fiction. This is actual medicine now, because in 2015, there was a virus called TVEC, which is a herpes simplex virus that was approved for use in melanoma, which is skin cancer. But it's also very clear that the last generation of viruses that we all designed weren't very good, okay? They just weren't strong enough because we were so afraid of hurting people and hurting patients that we designed the viruses to be very mild. And we knew it as we were doing the trials, because no virus that's been given to man so far has ever reached what we call MTD, maximum tolerated dose, in clinical trials. We've never been able to give enough virus to have someone get really sick, okay? So it isn't surprising that only one of the viruses in the last generation came back to be important. So in 2014, when I knew this was about to happen, that's why I came to the City of Hope. Not only because of its robust robotic programs and its offers for me to go lead a very big surgery program, it is also because at the City of Hope, there are 3 synthesis facilities that allow me to go, whatever we design that is good, go make it into medicine and to go put it into human testing as quickly as possible. So since arriving in the City of Hope, we've designed over 400 new viruses that may kill cancer. We've put these viruses through something called high throughput screening, which means we take a panel of cancer cells called the NCI-60. These are the 60 cancer cells that the National Cancer Institute believe are the most representative of the human cancer. And we've thrown these viruses on to see which ones killed cancer. Now, most of the time when we design cancer therapies in the last century, in the 20th century, we would say, oh, we want to design a lung cancer drug, or we want to design a colon cancer drug. In this century, I said, why don't we just change that? Why don't we just go find something that kills cancer? So our bar was quite high. We wanted to find those viruses that killed every cancer in the NCI-60, and we found a bunch of those. Bunch of viruses that killed everything in the panel, because then it potentially could be developed for use in many different useful cancers. Then we took those that killed everything and tested them in animals to see which ones were safe. We found a bunch of those. And so you'll hear about a virus that we're taking forward called CF33, and it's a 33rd virus that we tested. It kills everything in the NCI-60, and it is enormously safe in animals. We can give about a 5 to 7 logs higher virus than would hurt animals. And so that is actually the virus we're moving forward to human testing. We've also redesigned it to carry certain genes to help fight the cancer even better. So we have put into the virus, the first virus we're taking forward is a virus we have named CheckVax. And this is really the CF33 virus. It carries a gene called human sodium iodine symporter, HNIS. HNIS takes up iodine, so it is a normal human gene that's in our thyroid cells that take up iodine so that our thyroids can produce thyroid hormone. It also allows us to give iodine and be able to track where the virus goes. Okay, so these are PET scans showing virus going into the tumor by just simply tracking where the iodine goes. We also put into it an anti-PD-L1. This is one of the immune therapies that's currently used for triple-negative breast cancer, for lung cancer, for skin cancer. That's highly effective. So not only did we want a virus to infect and kill tumor cells, we wanted to leave behind immunotherapy and be able to be tracked. And this particular virus in animal studies have been quite remarkable. We are able, in this red line, to cure animals with just 1,000 particles of virus, okay? And that's about 3 to 4 logs better than anything that has ever gone to humans. And so we are in the midst of our submissions to the FDA. We're in the second round of doing that, and we're hoping to be in human clinical trials for triple-negative breast cancer, colon cancer, lung cancer, and skin cancer by the beginning of next year. We're also working on a different type of immunotherapy called CAR T cells. So let me explain what those are, okay? And T cells are a kind of immune cells in our body that protect us every day against many things. They protect us against viruses. They protect us against bacteria. They could also protect us against cancer if they could see the cancer as being foreign. So if you look at the cartoon here on the bottom right, here's a T cell. It has receptors on the outside, and this receptor finds an antigen, meaning a protein on the surface of something that is bad, and recognizes it as something bad. It will then, this T cell will go crazy attacking it and killing it. It's just that for most cancers, our T cells can't recognize it as foreign. The reason that they can't is because our body has protected cancer cells because our cancer cells came from us, came from self. And our body is so worried that our immune cells will start attacking us, I mean our own tissues, that it has a protective mechanism that also protects cancer cells. So what CAR T cells are really genetically engineered artificial T cells that recognize something that we know lives mainly on cancers. So we take a viral particle, we put it in to infect the T cell, then the T cell could express what we have encoded in the viral particle to be these hybrid receptors that recognize something on the top of cancer cells. So again, that allows us to attack the cancer cells and teach the T cell what to do. But from practical terms, It really is taking the T cells out of a human being, reprogramming it to go in and recognize the molecules on the surface of the cancer that we care about. We replicate that in the petri dish, and then we send it back in to attack cancer. Okay, and so again, very, very interesting thoughts of what to do, and in reality, oh, I'm missing a slide here. Hang on one second. I'm so sorry. Hang on one second. I need to go back. I'm missing a slide, but what that slide actually goes to show is this. Okay, this is not just, again, laboratory medicine or preclinical medicine. In reality, we are now using this as clinical medicine. That's because there is a molecule on top of a kind of cancer called leukemias and lymphomas called CD19. This particular molecule, we've made CAR T cells against it, and it actually brings remission in certain lymphomas and leukemias, even if all chemotherapy and bone marrow transplant has failed. And that's now standard therapy, okay? And the problem is finding other antigens on top of cancer cells of other types that we can actually use to treat, okay? So at the City of Hope, we've actually worked on an antigen based on a protein called alpha-fetoprotein that's in liver cancer. This particular protein is really important in liver cancer in the sense that 90% of a certain type of liver cancer that starts in the liver express it. And that type of liver cancer is called hepatocellular carcinoma, and that takes the lives of 600,000 people worldwide every year, and we have no good treatment for this type of cancer, okay? And we have now figured out how to make a CAR T cell that attacks it. This is in with a collaboration with a company called Eureka Inc. We've actually gone and tested it in human beings in China to start, because the incidence of this particular kind of cancer is much higher in China, and the results were quite remarkable. I'm just going to show you one case. This is Yulian Zhu, who is an oncologist in Beijing, and he had metastatic liver cancer that was in his bones and his lungs and in many places, and the alpha-fetoprotein level in him was very high, 67,000. Normal is less than 5. And he was in the process of getting sick and would have died within months. And by giving this CAR T cell to him, we were able to send him into complete remission, okay? Meaning all of the deposits of cancer were clinically killed. And so we have brought that trial back to America, and we're now in the process of doing it in a multi-center fashion in America. So again, trying to find new ways of using CAR T cells for treatment of human beings, and we are in trials here. But I'm going to end with this last slide, okay? But what about trying to find some way of making every cancer cell sensitive to CAR T cells, okay? So one of the collaborations that have been very, very fruitful for us is between me, Dr. Steve Foreman, who's head of hematology at City of Hope, and Saul Priceman, one of our best scientists. So we've gone and designed a virus, an oncolytic virus, so the same type that I had shown you before that can infect cancer and infect cancer in a very effective way and kill cancer. But into this virus, we actually put a gene for CD19. That other target I told you is now standard target for CAR T cells for leukemias and lymphomas. So by sending in the virus to do infection of cancer cells, not only do we kill some of the cancer cells, we make all of them express the CD19. And when we send in CAR T cells afterwards as a combination therapy, we've been able to cure animals with many different types of cancer. And this is one of the most exciting projects for us, because we believe this will allow us to be able to do CAR T cell therapy for almost any type of cancer that a human being can get. So we are actually in the midst of moving forward to human clinical trials. And again, we're hoping to be in human clinical trials either end of 2021 or the beginning of 2022. Okay? And so a lot of exciting things. We're still on mission moving things forward. I'm going to skip over the next slides because we are out of time. And but again, I really appreciate your being on the call today, and I just wanted that since this year we are not going to be able to get together at a luncheon or at a banquet, that we maintain this dialogue, because in order for us to do all these innovations, it requires trials, it requires laboratory studies, and we can't do it without you. Okay, and that's because the mission of moving all of this forward, unfortunately, it's a very expensive process, and so I am so appreciative of the real estate group and all that it's done through the years, and the $55 million you've raised for the City of Hope. We are trying to put all of it to great clinical use and to laboratory use to try to move medicines to man, to save lives, to produce more cures, and in a much more less invasive fashion. So I can't say thank you enough, and thank you for joining us today.
Thank you, Dr. Wong. We have a few questions for the question and answer session. First question, can you talk a little bit about heparin?
Oh, heparin for COVID-19. I assume that's what the question is. It's very clear that when patients get severe infections and get the immune reaction on top of it, many of the organs are dying because of clots that are forming within the microvasculature of the organs. And that's why when patients get severe infection, people are now trying to give heparin and heparin-like substances to try to declot those organs in order to preserve life. So a lot of those trials are ongoing, and in many ICUs, there's a whole protocol to go use it. And again, it's going to be a lot of things coming together to try to bring somebody who's very sick back. But it seems that a number of important ICU groups are putting that together.
Thank you. Dr. Fong, if you have had a flu shot and pneumonia shot, does it help to prevent getting COVID or help fight COVID if you get it?
The answer is probably no. Okay, and the reason is the reason you have so many different vaccines. Okay, well, why do we need vaccines for mumps and measles and polio, and it's because, again, each of the viruses that we are faced with in nature could act very differently. Okay, and finding out what is immunogenic in that virus and how to go make a vaccine for it. Not so many of the vaccines for one, most of them will not work for another. Okay. In fact, year to year, flu vaccines don't work, right? And that's why we need a flu vaccine every year, because the strains change. Viruses live to change, okay? And they're constantly trying to pick up genes and shed genes that will allow them to live better. In fact, one of the big worries right now is that it's very clear that the coronavirus-19 virus is evolving, that the strains that are seen in Europe are very different than the ones that were seen in China. And as it comes to America, depending on where it lands, could be a different strain. So some early data, for example, from Arizona tells us that's closer to the ones in Europe than the California strains, which are closer to the ones in Asia. So will we need to make multiple vaccines in order for even to treat COVID-19? So these are the worries, and but again, I probably won't protect you, but I say probably because a robust immunologic reaction is always good, and having other components of the immune system primed already, theoretically, when folks have been getting vaccinations, get other vaccinations, it should work better and faster.
Our next question. With 150 companies working on COVID vaccinations, are there any of them sharing data and collaborating?
Yes and no. Okay, so you need to know, and I'm going to try to be very politically correct here. Okay, and so it's been amazing how collaborative most people have been. Okay, so right now you can actually go on the internet and pull down all 200 full sequences of COVID-19 that have been completely sequenced worldwide. That's because most of the scientists and many companies have decided that providing all the information possible allows us to move fastest. Okay, so that's why there's so much data. Okay, we wanted to figure out where on the spike protein we should target, and we were able to go on the internet and find a huge amount of data that other people spend long hours working on, but they were willing to share. But whether there will be complete sharing of data going forward, again, depends on company from company. But overall, I've been very impressed with how collaborative most people have been.
Thank you, Dr. Fong. Can you get infected with COVID twice, or do we know this yet?
Yes. It's very clear that let me backtrack. You can get the disease more than once, okay? So for some folks, it could just be the virus sticking around and the body can't get rid of it, and then they become symptomatic again. But it's also very clear that people can get reinfected. Folks are now trying to figure out why it happens. Is it just because antibody levels go down and they are just infected with the same kind of virus, or is it because they are now exposed to two different strains? And what are the determinants of somebody at risk for getting it again or getting it reactivated? Meaning they got rid of it, the body got rid of it the first time, and now it's come back.
All right, another question for you, Dr. Fong. Is it safe to have screening and appointments at Browerman?
Right now, it's been very, very safe, okay? And we've asked patients not to come if they're sick, and by and large, that's what everybody's complied to, okay? We have complete screening. You can't actually enter our facilities without going through a screen, and that screen for temperature. And we do not allow visitors except for very, very specific indications. And so somebody clearly disabled, both either physically or from a mental capacity standpoint. And so we've kept the clinics very, very safe. And if you've come to the clinics, you would see that our sitting areas are set up for social distancing. We are now scheduling according to social distancing. I'm not allowing members of my faculty to double book into the same slots to crowd up the waiting area. So it's been very safe. And so far, so good. But that's also where I will ask for your patience, okay? Meaning that if you knew how many times I get emails or calls a day to ask for exceptions to the visitor rule, and most of the time I say no, because protecting our patients is one of our highest priorities. And so the other way we've done it for families is that we've now set up for web meetings and video to allow us to be able to provide information for the family, even though they can't physically be present for the initial office visit. So those televisit videos have been invaluable in making sure that everybody stays on the same page for treatment of cancer for their loved ones.
Excellent. Let's ask a yes or no question to end the session. Is it safe for students to go back to school?
It depends on how it's set up at school, okay? And that's why a one-size answer is not right, okay? And that's because it all depends. So some schools that I teach at two different schools here in Los Angeles, and one of the schools has decided they're going to bring the students back and they're going to go into a 14-day quarantine. Then they're going to set up the classroom so that no one will actually sit more than within 6 feet. So they put in a lot of safeguards to go do this, and they've actually going to institute very rapid testing for the kids. And so it all depends on how it's set up. And that's because we can't stay in a bubble forever. And if we just are all responsible to each other, okay? When we're out, wear the mask, and that's because it really is important to protect each other and to do the social distancing where you can. We need to get back to normal life, and trying to make sure that we go do it safely, really important, okay? And that's why now one of my hashtags on my Twitter is hashtag safe and social, okay? And that's because it's not either or. It's how we all work together so that we can actually all get back to life. And obviously, getting back to healthcare is really important.
Excellent. Thank you. Well, in closing, I'd like to thank Dr. Fong for his time today and for all the great work he and his team are focused on in our fight against cancer. We thank you as well to our generous sponsors, Langen, Perkins & Will, TMG Partners, and the San Francisco Business Times, and frankly, to all of you joining us here today. We plan to continue this series with sessions in September and November, so stay tuned for those dates and those subjects. If you and your company would like to support Dr. Fong's incredible work, please do text. It should be on the screen for you right now. City of Hope Rec, or COH Rec, to 41444. Any and all support is, of course, appreciated, and I can promise you these go directly into the treatments and research. If you'd like more information about City of Hope or to get involved, email our City of Hope staff team at sfcityofhope@coh.org. And we thank you. Look forward to seeing you in September. Thanks, everybody. Thank you, Doctor.